Last class we talked about Drosophila, fruit fly sex determination. We talked about Caenorhabditis or nematode sex determination. And then today we get to move on into some details about humans. Remember, the focus last class was about how mutants tell us something about how sex determination works. So maybe it's worth starting there. So what are the genetic sexes of males and females, or hermaphrodites in the case of C. elegans? So what are the sex chromosomes in flies? Fly males have what sex chromosomes? They're XY. And then the females in fruit flies, Drosophila, have what sex chromosomes? What goes next? They like humans? Well, they're like humans in some ways, but not in other ways. So how many X chromosomes does a Drosophila female have? 2. Looks like human, right? What we're going to do today, by the way, is we're going to add humans to this table. Hey, let's start there. Humans, males, XY. Females? Come on. 2 X's. So what's the difference between human and fly sex determination?
The X determines the sex of the fly.
Okay, so what we talked about last time was that in fruit flies, it's the number of X chromosomes that tells you what sex you're going to be. If you have 2 of them, you're female. If you have 1 of them, you're male. What is it that does sex determination in humans? Is it the number of X chromosomes, or is it the presence or absence of the Y? That's what we're going to learn about today, is that the Y chromosome is the thing that causes maleness in humans, and without it, you're female. So what about worms? C. elegans. Females? What sex? Or what sex chromosome constitution? Anybody? What sort of chromosomes do female or hermaphrodite C. elegans have? Which sex is it that's XO? One of the sexes has 2 X chromosomes, one of the sexes has 1 X chromosome. So the male has 1 X chromosome, and then nothing. So it's not a chromosome. The O is nothing, represents 0, essentially. And then the hermaphrodites have 2 X chromosomes. Like in Drosophila, in Caenorhabditis, in these nematodes, it's also the number of X chromosomes that controls sex. Clearly, there's no Y chromosome, so the Y chromosome can't do anything in sex determination in C. elegans, in the worm. So we're going to start class with an example of temperature-dependent sex determination. We're going to switch back at the end to talking about human sex determination and comparing that with fruit flies. So in that table we just drew, male Drosophila and male humans are both XY. Female Drosophila, fruit flies, and female humans are both XX, but there's an important difference in how they do sex determination. That's one of the goals for today, is to be able to understand how mutants told us something about those sex chromosomes and how sex determination works. And if we have time at the end, we'll talk a little bit about behavioral genetics, which marginally has to do with sex determination. Remember, last class I talked briefly about clownfish and how social sex determination works, that by being able to sense the genders of the people around you, that can sometimes, in certain species, tell you what sex to be. Here's another example, temperature-dependent sex determination, which I mentioned briefly last class, and some of you said that's a common form of environmental sex determination. The temperature that the eggs incubate at dictate the sex. So red-eared slider turtle, for example, lives on the East Coast. If the eggs develop below 28 degrees, everybody becomes a male. And if it's above 31 degrees, everybody develops as a female. So there's a sweet spot, this temperature in between, where you get roughly 50-50. So 50% female, 50% male. So this is critical temperature for the survival of the species, as I mentioned briefly last class, because if you get too hot a temperature or too cold a temperature, you don't have individuals of the other sex to mate with. So what makes, in this case, what makes sex determination dependent on temperature? How can temperature affect an organism? Pardon?
Metabolism.
Okay, so it could change metabolism. So
the enzymes and the hormones that are being used.
okay, so it could have something to do with enzyme activity.
Right.
So at hotter temperatures, some enzymes are more active. At hotter temperatures, different enzymes might be less active. You get them too hot, sometimes enzymes stop working. So how would that impact, how would enzymes, and you mentioned hormones too, how would that impact sex determination? We're heading in the right direction for this story. So temperature, hormones, enzymes. It turns out that testosterone and estrogen, or estradiol in this case, are actually pretty closely related compounds. And there's a single enzyme that converts testosterone into estradiol in this species, one enzyme, aromatase, right here. So it's a single step. There's one enzyme that converts testosterone into estrogen, basically. And of course, this impacts sex determination. So if you're a female, do you want your aromatase, if you're a female embryo, do you want your aromatase enzyme active or inactive? You want to produce estradiol, so if you're a female, you want aromatase to be active. And if you're male, hell no. You don't want your testosterone being turned into estradiol, you want to keep it. So in males, that enzyme is inactive. Right? Testosterone gets produced as a part of a biochemical pathway, and that's the end point in a male. In a female, there's that extra step of aromatase converting testosterone into estradiol. So you'd expect that the aromatase, in the case of this hypothesis, the aromatase enzyme would have a higher activity at a higher temperature, and that would cause the production of estradiol from testosterone, and at a lower temperature, that this enzyme would be less active. Hypothesis and an expectation. And it is, in fact, what scientists observe. So they extract aromatase from male embryos, female embryos, or presumptive male and female embryos at the two different temperatures, and they find that, indeed, the aromatase activity is higher in ovaries at a higher temperature, and at a low temperature, you get low activity of this enzyme. So this is a great molecular mechanism for understanding how temperature could impact sex determination. You have enzymes that are sensitive to temperature. There are enzymes involved in producing hormones that are important in sex determination. So there's a pretty clear mechanistic link there between environment and what's going on inside an organism. Any questions about this concept? It's just one example of how temperature can affect sex in one species. just wanted to give you one case, one piece of information about how environmental sex determination can work. So absent any other questions, we talked about humans and human mutants and how human mutants told us how sex determination occurs in humans and how that's different from what happens in Drosophila. And the really cool thing about humans is they go to the doctor, which Drosophila don't do. So it's really hard to find these Drosophila mutants that were used to figure out how Drosophila sex determination worked, because the scientists had to sit there and look through thousands and tens of thousands, probably fruit flies, until they found ones that had disorders of sexual development. But in humans, if 99% of live births are maybe more like 99%, 1 in 100 individuals doctors report are not clearly morphologically one sex or the other. So about 1 in 100 babies, it's not clear whether or not they're completely male or completely female anatomically. So about 1% of live births are hermaphroditic, somewhere in between, in that continuum between male and female. So this is useful for studying human sex determination, because when those things happen, they get reported in literature, they get reported in physician records, and those can be studied. So normally, of course, we have XY males, as we discussed at the start of class, and XX females in humans. Please tell me, is it the number of X chromosomes or the presence or absence of the Y that causes sex in humans? Is it the X or the Y? The Y. How did we figure this out? What did we talk about last class? We can't do these sorts of experiments on humans, but what sort of mutations or what sort of experiments would you do genetically to figure out if it's the Y chromosome that causes maleness? So we're back to this concept of necessity versus sufficiency. So if you want to show that the Y chromosome is necessary to be a male, what sort of a genotype of individual would you want to look at to find out what the effect of the Y chromosome is on sex? What genotype? Right. You'd want to have an individual that's got a normal set of autosomes, so diploid on all the autosomes. I'll say AA, autosomes. They have 2 copies of every autosome. They have an X chromosome but no Y. Also no X, because that would be a female. We know that XX is female. What about sufficiency? XXY. Right. So then you want an individual that's diploid for all of their autosomes, but 2 X chromosomes, that would be female. If the Y is sufficient to make you a male, then you just add it into a normal female genotype. You just add the Y chromosome. 2 X's plus a Y, what does that do to sex? Is the individual still female, or is the individual male? Does anybody know what syndrome XO humans have? This is Turner's syndrome, individual, and this is, again, getting back to this idea of how humans are such a powerful genetic organism, because we go to the doctor, and the doctors collect all this information, they name syndromes. So individuals that are lacking an X or a Y chromosome, they just have one sex chromosome, are individuals with Turner's syndrome. And then XXY individuals have Klinefelter's syndrome. That's what we'll talk about for the next few minutes. Okay. So are Turner's syndrome patients, make a prediction, are Turner's syndrome individuals going to look female or male? What do you think? It depends on how we do sex determination. Are we counting X chromosomes, or are we looking at presence or absence of the Y? Y. So if you are XO, you don't have a Y chromosome, what sex would you generally look like, male or female? Yeah. So I'll say approximately female. Okay. What about Klinefelters? The Y chromosome causes sex determination in humans. So individuals with Klinefelter syndrome are approximately male, with a lot of caveats. So phenotypically, external genitalia and so forth, phenotypically more male than female. How is this different from Drosophila? So in black here, what would an XO Drosophila, what sex would they be? So 1X, maleness. So this is the critical difference between the genetic sex determination of Drosophila and humans. Both of them have XY and XX sexes. But in Drosophila, an XO individual is male, and in Drosophila, an XXY fruit fly is what? Female. 2 X chromosomes. And that's when we learned that fruit flies are not humans. The end. Well, maybe just one more nail in the coffin. Questions or clarifications on this? Okay. So individuals with Turner syndrome, relatively rare, about 1 in 2,500 live births. There's a characteristic set of morphological features: small neck, broad shoulders, a little bit of webbing sometimes between the neck and the shoulders. I was talking to a genetic counselor one day, and she told me that you can practically diagnose individuals with Turner syndrome over the telephone. You don't even have to see them. You can ask them some questions and figure out whether or not they likely have Turner syndrome. And that has to do with this bottom point about failing high school geometry. Turns out that individuals with Turner syndrome and Klinefelter syndrome have an interesting set of neurological differences between the average XX or XY individual. So before we get to that, though, I just wanted to show you this for comparison with Turner syndrome. Klinefelter syndrome is a little bit more common, about 1 in 600 live births. And they do have some feminization, a little bit of breast development sometimes, but penis, testes, yes, smaller usually, and some other phenotypic differences. Turner syndrome and Klinefelter syndrome patients are often sterile, which may or may not be a surprise. So it was the external morphology of these individuals that provided that information about, yes, Turner syndrome XO are mostly female, not entirely, and the same then for Klinefelters. Add a Y chromosome to an otherwise female genotype, an XX individual, and then you get development of a penis, testes, etc. So that showed that the Y chromosome itself is enough to cause those sorts of changes from a female XX body to a male XXY body. Add a Y chromosome and you induce these changes. The Y is sufficient to cause some parts of maleness. And I know this is small, so hopefully you've got it and you can zoom in. There's an interesting survey paper, summary paper review published a few years ago about these cognitive and neurological differences between people with these sex chromosome differences, XO and XXY. And one that's particularly of interest, again, for diagnosing Turner syndrome, is right there. Visuospatial differences. So most people that have Turner syndrome have issues, and this is one of the things that this genetic counselor told me when I talked to her about Turner syndrome patients. She said the worst thing you can do to somebody who has Turner syndrome is to give them directions. drive down the street half a mile, turn right, at the next stoplight, turn left, have a hard time visually imagining that series of directions. So they often fail geometry or do really badly in geometry. One of the least good, one of the worst subjects they usually have. And this is the way sometimes in conversation you can tell if somebody has or is likely to have Turner syndrome. It also says under the arithmetic category there are difficulties with subitizing, which is the ability, if you don't know, the ability to look at a group of objects and approximate how many items there are. That's that process. That's also something that a lot of people with Turner syndrome have a deficiency in. Like estimating large groups of objects, how many are there? You know, that thing where they fill the jar with M&Ms and you have to guess how many there are in the jar. Yeah, difficult. You'll notice if you go down a little bit farther, we get to speech, and then down here at the bottom, social and psychiatric differences between XX, XY, and then Turner syndrome and Klinefelter syndrome. So there's some other subtle differences that having the wrong number of sex chromosomes makes in individuals. And that brings us to some of the more interesting, I think, biological aspects of having a non-normal number of sex chromosomes. Like whether or not XYY supermales exist. What do you think? Get two Y chromosomes? I don't have two Y chromosomes, I'm pretty sure. What happens if you have two Y chromosomes? These individuals exist, XYY.
More aggressive.
So why would they be more aggressive? I agree. So, you talked about it last semester? In which class?
Dr. Schreiber?
Yeah,
Yeah.
cool. So, why would an extra copy of Y chromosome make you more aggressive? Yeah, so maybe there are genes on the Y chromosome that make more testosterone, maybe. Not necessarily the case, but this was a hypothesis that a long time ago caught hold. And of course, all science starts with hypotheses, and it seemed a reasonable one at the time. So we jump back to 1965. This was published, Christmas, 1965. Aggressive behavior and mental subnormality in the XYY male. So there's this hypothesis, and this had come from earlier research. 1% of males in institutions for the mentally subnormal have an XYY sex chromosome constitution. So basically 1% of incarcerated males, or something like that, are XYY. What do you want to know? What's the first question you should be asking when you read that? Yes, sir?
It says XXY.
Sorry, XXY. We're going to, yes, you're right, XXY. Klinefelters. Sorry, we're not quite at XYY yet, but we'll get there. But what do you want to know about this statistic? So the point is they're studying people that are incarcerated, and there's a hypothesis that maybe these individuals have some difference in aggression or the tendency to go on crime sprees, and that therefore there's more people in prison that have sex chromosome aneuploidies. XXY, for example. If this was a commercial and it said 1 out of 10 dentists recommend this toothpaste, what do you want to know?
How many genders?
Okay, so how many dentists did you ask? Did you ask 10 dentists and one said, yeah, I like this toothpaste, and the other 9 said that one? So what you might want to know is 1% representative of the population? That's the comparison you have to make. So big deal if it's 1% of a prison population. What was the incidence of Klinefelter syndrome? A few slides back. It's like 1 in 1,600, about 1 in 1,000. So that might make the argument, if there are 1 in 1,600 or 1 in 1,000, let's say on average, people in the population that have Klinefelter syndrome, and it's 1 in 100 in prison, then it looks like there's an enrichment in prison for people that have Klinefelter syndrome. So that's what this study started to look at, was they wanted to study whether or not this was a real correlation or if it was just chance. Later, 1968, so 3 years on, another group basically does a similar study. Tall criminal American males, incidence of gross chromosomal errors. So they find that 1 in 10 subjects, in this case, had some sort of sex chromosome aneuploidy. Pretty striking, isn't it? 1 out of 10 inmates that are male and tall have Klinefelter syndrome, or here we go with the supermales, 47XYY. They were all mentally ill, and none had been cytogenetically diagnosed. That means they just went into prison, they said, give me your tall men. And then they took samples from them, and they looked at their chromosomes, and they discovered that basically 1 in 10 of them had some sort of sex chromosome aneuploidy. So what do you think? Is there a link between your sex chromosomes and your propensity to commit crimes and be put in prison? Seems like it. We're enriching, it looks like prisons are enriching, they're condensing males that have sex chromosome aneuploidies. And then this. So what are some arguments against this? Right? There's always 2 sides to a coin. So the crimes did not generally involve aggression. And they actually said maybe it's not aggression at all. Maybe it's that there's a reduced IQ in people that have sex chromosome aneuploidies, and that they get caught more easily, and that's why they're in prison. It's not because they're more aggressive or more likely to commit aggressive crimes. It's because they're more likely to get caught, right? There's always an alternative hypothesis you could formulate. There's always another explanation. By the way, I got all this from Wikipedia. Wikipedia is okay, as long as you cite it. At least I got all these citations from Wikipedia. It's a great place to find links to the literature to read. Not great for actually writing papers on, but here's the most recent conclusion about this. XYY males, 2012. These authors suggested that maybe it's not anything to do with the presence of 2 Y chromosomes. It has something to do with the environment of the individual, the actual social environment. So risk of conviction was moderately increased, but was similar to controls when socioeconomics was taken into account. So it wasn't so much that it was whether or not you had an XYY genotype that landed you in prison. There was a correlation between whether or not, and this is the weirdest part, I think, there's a correlation between whether or not you have sex chromosome aneuploidies and your socioeconomic status. So if you were poor, it seemed like there was a correlation with likelihood of having XO, XYY, XXY, and other disorders of sexual development on the chromosomal end. Isn't that interesting? So what might be the link between your socioeconomic status and your genotype? That's area for new research. So in other words, to wrap all this part about sex chromosome aneuploidies up, the state of the field now is that there's no correlation between aggression and sex chromosome aneuploidy. XYY supermales aren't necessarily enriched in prisons. It might be due to other factors that don't have to do with their genotype. talked about this point a little bit. What does an extra Y chromosome have anything to do with hormone levels, anger, socioeconomic status, or any of that? It's unclear, and that's why this original hypothesis from the mid-60s hasn't really held up when people started doing controlled studies. There's no intellectual justification for why an extra Y chromosome would have any of these effects necessarily. So at this point, scientists think it's basically correlation, not causation. Like the fact that increasing cheese consumption in the US is correlated with your likelihood of being strangled to death by your bed sheets. Pure correlation. The fact that the trends are similar has nothing to do with the fact that eating cheese causes you to die in your bed. All right. One last quick point, more for fun than anything else, hopefully. Thrill-seeking in beetles. So I wanted to make one last pitch for how genetics not only impacts everything we've learned about this semester, but also behavior. And this is one of the more interesting papers I read recently. Thrill-seeking, the influence of mitonuclear genetic variation on personality in seed beetles. And I read that title, and I said, what the hell is personality in beetles and how do you study it? So the title made me open this PDF and read what the hell they're talking about. How do you measure personality in beetles? And what they're talking about here, mitonuclear genetic variation, just means that there are mitochondrial chromosomes. So each of our mitochondria in each of our cells used to be a bacterium until our ancestral cells enslaved them and turned them into our little batteries that produce ATP, chemical energy, to run our lives. So mitochondria have circular chromosomes. So those are inside the mitochondria. And then over here in the nucleus, we have our nuclear genome. Pairs of different sized chromosomes. We've talked a lot about that in class. So we have our nuclear genome and we have our mitochondrial genome. And most of the time we get our mitochondria from our moms. The mitochondria are maternally inherited. And then we get half of our nuclear genome from one parent and half from the other. So every time we have offspring, mom contributes her mitochondrial DNA, and then mom and dad equally contribute the nuclear DNA, 50-50. And what they're talking about here with mitonuclear genetic variation is that there's another genome besides the one we normally talk about in genetics, the nuclear genome, the autosomes and the sex chromosomes. Our mitochondria have genes too. And apparently that can play a part in a number of aspects of life, including our personality if you're a seed beetle. So seed beetles, you can measure, what they meant by personality is whether or not they are active or inactive. So whether or not beetles are more or less likely to go roaming around in areas of, say, imagine you're a cockroach and you're living in a human's house. You tend to stay along the sideboard or the floorboards, right, or along the baseboards, around edges, underneath toe kicks in the kitchen, behind toilets, that sort of stuff. So basically what they're doing here is they're measuring whether or not these beetles are more likely to be found out in the open, running around, or somewhere under cover when they do these tests of personality in seed beetles. So here's what they did. They took a bunch of these beetles, and they set up different crosses so that they combined different nuclear genomes up here. So they've got nDNA is nuclear DNA. So these are the nuclear genomes. So they have different seed beetles that come from Brazil, they come from California, they come from Yemen. So same species, just collected from different parts of the world. So the circles, squares, triangles, 3 different nuclear genomes. And then what they do is they make combinations of those, as you see down here on the bottom, with 4 different mitochondrial genotypes. So mitochondria from Brazil, 2 different species, or 2 different populations from California, and then Yemen. And here on the y-axis, we've got a scale of activity. So how active are these beetles? How likely are they to be found out in the open, running around, as opposed to cowering in the corner? So it's like cowering is up there and thrill-seeking is down here. Right, highly active at the bottom, inactive at the top. So just to orient you on how to read this, let's look at the circles first. So these are females, the filled-in circles, for example, and filled-in triangles and filled-in squares. Those are the females. So if you take a female, so it's circular, female. Circular is Brazil, sorry, filled-in female. Brazilian nuclear DNA over here, and she's in the column with Brazilian mitochondrial DNA. So that's a typical control individual. It's a Brazilian seed beetle. Relatively inactive. What happens if you replace her mitochondria with mitochondria from a different population, California or Yemen? Does her activity change much? Not really. It changes a little bit, but then the error bars are pretty big too. So basically, this data series says it doesn't matter whose mitochondria you have. It doesn't matter which mitochondrial genome flavor, version, allele you have. It doesn't change your behavior. Where is an example of this on this chart that mitochondrial genotype does influence behavior? That is, when you compare the 4 different columns, where do you see drastic changes in activity? So males, Brazilian. So we're looking down here on the bottom, we're looking at circles. That's the Brazilian nuclear genome. So if you're Brazilian mitochondria, Brazilian nuclear genome, you're somewhat active. But then what happens if you're Brazilian with Yemeni mitochondria? Now you're even more active. So all this says is that your mitochondrial genome, whose mitochondria you have, can influence your behavior. Right. Same species of beetle, same nuclear genome from Brazil. The only difference between those two that are circled in blue is which mitochondria they have. The Yemeni mitochondria or Brazilian mitochondria from this species. And just making that one swap, taking out one mitochondrial genotype and putting in another one from the same species makes an effect on behavior. Why would that be the case? Come up with a story, a hypothesis that might explain or might suggest why mitochondria has anything to do with behavior? What do mitochondria do? They make ATP. So can you correlate that somehow in a hypothesis or a story with what we're seeing here?
More mitochondria, more energy.
So it might be that, sure, so it might be that this combination of nuclear genome, nuclear chromosomes, and mitochondrial chromosomes makes more ATP. So your cells have more energy, so you feel more energetic. And then you find somebody to synthesize this and you sell it on infomercials and you make a ton of money. Getting ready to study for finals? Yemeni mitochondrial DNA from seed beetles. It's been, okay, maybe not. I'll have to work on my pitch. You'll see it on TV. So that could be what's going on, is that different mitochondrial genotype affects ATP production, and that maybe those individuals that are thrill-seeking make more ATP. It's so simple and elegant, it's likely to be wrong, but it's a hypothesis, and it's worth testing. So hopefully everybody will be comfortable next time they hit Pike Place Market and they have to figure out which bathroom to go into. But this raises a really important point that's really in the news currently about unisex bathrooms, or how you identify bathrooms as for use by individuals of, well, there aren't just two sexes. It's a continuum. So how would you better label bathrooms now that you know a bit about sex chromosomes and sex determination? What's a better way to label these bathrooms? this is basically saying male and female. What do you think is maybe a wiser way to label bathroom? Should we just have no more, choose this bathroom or that bathroom? What do you think? Impact of genetics on society. Any thoughts? What if we did this? Should we just do you make sperm? This bathroom. You make eggs? That bathroom. Does that work? What if you're sterile? Or what if you're a hermaphrodite? Just one bathroom for everybody? Okay. Go for it. We'll see what happens. But pay attention to the news because there are a lot of laws and lawsuits right now going on about bathroom labeling. North Carolina, is it right now? Or Virginia? I think they're, check CNN. You got tablets, CNN.com. So that's the end of content for the semester. We've talked about transmission genetics, we've talked about molecules of DNA, we've talked about how DNA replicates, transcription, translation, mutation, and now we move on to next class, which again is the review session. For those of you that haven't worked on this yet, this is quick question. Don't spend too much time writing on it. You could if you want, but don't kill yourself over it. I'm interested in you reflecting a little bit on what we talked about in class today. This is the last points for the semester. There will be maybe a 1-point thing on Wednesday, so do come to the review session. We're still doing in-class participation points, but it'll be mainly review session on Wednesday, and then on to the exam.